Good morning, students. Today we are going to study about the HPLC. HPLC, it is nothing but what? High performance liquid chromatography. It is a one type of chromatography and it is called as high performance liquid chromatography. And it was first discovered by Horvath in the year 1974, and HPLC has expanded very quickly. That is, HPLC is an abbreviation for high performance liquid chromatography. It has also been referred to as a high pressure liquid chromatography. HPLC is a type of liquid chromatography in which the mobile phase is liquid that flows through a reusable column by means of pumping system used to separate compounds that are dissolved in solution. HPLC is a very efficient technique and can be applied to about every kind of sample. It yields excellent separation in a short time and can work with minimal training of the operator. Today, HPLC is one of the most widely used technique for separating and analyzing mixtures of chemical substances, compounds and material. So it is a one type of the chromatography under the chromatography, and it is the advanced chromatography. So within all the chromatography it is the advanced one, and it is a diagram here showing the chromatography technique. So scientists realized that the major increase in column efficiency could be brought about by decreasing the particle size of packing to increase the surface area and thus interaction sites and retention. So this required sophisticated instruments with high pressure pumps to assure reasonable flow rate through the very narrow pores. So here the instruments required are the pump and column chromatography and injecting for the injecting sample injection and solvents. So this is a principle of liquid chromatography here showing first of all the column is there, and in that column stationary phase is there. In second one we are going to injecting a sample, and in third column here showing the solvents are adding here, and in the next column the different separation of the mixture is taking place there. That is a blue color separation, green color separation and red color separation, like that. And in the last one, after the separation of each color of the sample, the mixture is separated there, and that we are going to collect it in the collector and analyze or collect the components here. And the components of the samples are separated from one another by the column packing that involves various chemical or physical interactions between their molecules and the packing particles, and the separated components are analyzed or collected at the exit of this column and identified by an external measurement technique. Separation is achieved by a different for the mobile and stationary phase for the components to be separate. So these are some separation mechanism comparison with the HPLC. So this is not for your syllabus, but what are the different instrument we are going to be used in the HPLC? You see here HPLC system, that is the main physical component, schematic of an HPLC system. So this is an HPLC instrument. So mobile phase solvent reservoir is kept in upper side here in the bottle shaped structure, and below that the machine is there. So in that machine, this is a setup of HPLC. Sample injector, auto sample sampler is there. In that we have to inject the sample, and below that the pump is there, and beside these column compartments are there, and lastly here the side of this HPLC the detectors are there, and here lastly after the detection of the samples or mixtures the control system, that is computer, is there. In that the data are available for us. So you see here in detail structure of this. sample syringe upper side, sample trays are there, and beside that column oven is there, column, injector and sample loops, pump heads, mixing vessels and mobile phase tubes. And next, mobile phase, this is a what diagram that is in the inside the machine how it is. Mobile phase that containing a solvent reservoir, and pumping system is there. Through that pumping system this solvent reservoir moving upside, and sample, after that we have to inject the sample, and then in the column this sample is going to be insert or moving in the column stationary phase. Next, through the whatever the sample comes outside, through that through the detector it will detect it, and after that in the computer the data acquisition and control system will takes place. So this is what the system or the setup of the HPLC, and the different parts are going to be used. They are solvent reservoir, the mobile phase reservoir can be clean, inert container, storage of sufficient amount of HPLC grade solvents for continuous operation of the system. It could be a single element, but usually multiple solvents are used in HPLC, that is a binary, tertiary and quaternary. Solvent reservoir should have a cap that allows for a tubing inlet line which feeds mobile phase to the solvent delivery system. The cap also serves to keep out dust, reduce solvent evaporation. So allow for pressurization of the bottle, or offer pots for the additional inlet lines and sparing. The material of the mobile phase reservoir depends on the type of application. Glass is typically used to prevent leaching of materials from the plastic into an effluent, which may be interfere with the analysis for RNA, protein, and electrochemical analysis. Plastic mobile phase reservoirs are often used, and a glass container can leach trace ions interfering with analysis. So this is called as a solvent reservoir where we have to allow the solvents. Next, mobile phase. The mobile phase is one or more solvent used to solubilize the components and move them in different rates across the column and the system. In contrast to gas chromatography, the mobile phase in liquid chromatography participates in the interaction with both solutes and stationary phase. Therefore, it should be carefully selected. Selection of the suitable mobile phase mainly depends on the application and the interaction mechanism. All mobile phase solvents should be freshly filtered and preferably degassed online, that is degassers, which are primarily used to remove small gas bubbles and reduce dissolved air. So this is a picture of showing the mobile phase, and this is some criteria about the mobile phase. And next instrument is pump, a solvent delivery system. The function of the solvent delivery system is to deliver the mobile phase. The solvent delivery system comprises the pump, check valves, flow controllers, pulse dampeners and pressure transducers. Delivery of the mobile phase must be pulse free to ensure minimal baseline noise for the pump. Next is injector. Injection valves are connected between the pump and the column and as close to the top of the column as practically possible. This allows an introduction of the analytical mixture into the stream of the mobile phase before it enters the column. An interchangeable sample loop of discrete volume is connected to the valve and isolated from the flow of mobile phase. The loop is filled with sample solution and the valve is then turned manually or electronically so that the loop is connected into the flowing mobile phase. Most modern injectors are autosamplers which allow programmed injections of different volumes of samples that are withdrawn from the vials in the autosampler tray. So these are the some syringe needles which are going to be used for injector. And next is the column. So the column plays a very important role here. So this is the heart of the HPL system, you can say. It actually produces a separation of the analyze or analytes in the mixture. Most of the chromatography development in recent years went towards the design of many different ways to enhance the column's properties and efficiency. So these are the different types of the columns which we are going to be used. So column dimension length is there. So different it affects both the efficiency and the speed of the separation. Longer columns have high plate numbers and yield better resolution, but with the longer analyze time. Note that column pressure drop is also proportional to the column length. So this is what some uh, the explanation about. Or next is stationary phase. So it is usually the smallest part. The column is the most important part in any HPLC system. The column is the only device in the HPLC system which actually separates an injected mixture. Column packing material are the media producing the separation, and properties of this media are of a primary importance for successful separation. The selectivity, capacity, and efficiency of the column are all affected by the nature of packing material or material of constructions. Since materials are polymer, science are highly rich in options, reactions, and modifications. Therefore, stationary phases faced various developments and can be still improved. So there are different types of the packing materials are there. So type of packing materials, that may be a non-particulate, porous, non-porous, core shell, packed, open tubular, like that. And type of base material is silica, polymeric, alumina, and zirconia. Stationary phase geometry is about surface area, interaction sites, pore size or diameter, pore volume, pore size, distribution, permeability, particle size, particle shape, and particle size distribution. Surface chemistry, type of bonded ligands, functional groups, bonding density, carbon content. And stability and rigidity is surface reactivity, chemical stability, physical stability, mechanical stability, stability under pressure, stability in common liquid chromatography solvents, pH stability, stability to hydrolysis in acidic and basic media, stability at elevated mobile flow rate. All these parameters are interrelated in their influence on the chromatographic performance of the column. The quality of an HPLC column is a subjective factor which is dependent on the type of analytes and even on the chromatographic condition used for the elevation of the overall quality. So this is some shape of the silica, irregularly spherical silica, monolith silica. And lastly, detector. So chromatography was discovered late in the 1890s. Its development was almost negligible until 1940, and this was largely due to the lack of an line sensitive detector. The detector measures a physical parameter of the column effluent or of components in the column effluent and transforms it to an electrical signal. The detector must respond sufficiently fast to the changes in concentration or mass flow in the effluent, otherwise, the peaks are distorted. Detectors can be classified into two types: universal and selective. Universal detector measures some bulk physical properties of the element, and selective means measures some physical or chemical properties that is unique to the solute. Detection may also be classified according to whether they are destructive or non-destructive. So these are the some common HPLC detectors. And lastly, data acquisition and control system. Computer based systems are used in every instrumental modules and at every stage of analysis. Computers controls the flow rate, element composition, temperature, injection volume, and injection process. Detector output signal is converted from analog form into a digital representation to recognize the presence of peaks, and then at higher level of computer analyze a chromatogram gram is obtained. So here you see here on the screen of the computer how here this shows the peak, like how we are using or drawing in the graphs. So the depending upon this peak we are going to separate the different components. So these are the some factors which are influence the HPLC. And application of HPLC. So here field of application is in the pharmaceuticals we are going to be use or separate the components like antibiotics, sedative, steroids, analgesics, drugs and metabolites. In biochemicals, amino acids, peptides, proteins, carbohydrates, lipids. In the food products, artificial sweeteners, antioxidants, preservatives and vitamins. In the industrial petrochemicals, condensed aromatics, suffocants, propellants dyes, crude oil, characterization of petroleum products, hydrocarbons, forensic chemistry, drug, poison, blood, alcohol, and narcotics, clinical medicines, bile acid, drug metabolites, urine extracts, estrogens, and stimulants, toxicological, drug screening, systematic toxicological analysis, environmental and pollution.